In our previous lectures, we've seen how the unique collection of proteins that characterize any particular cell comes about as the result of highly regulated mechanisms for controlling the expression of genes, and also as the result of mechanisms for delivering the protein products of those genes to the right place. Of course, it's by having different sets of proteins that cells are able to be specialized for different kinds of functions. Or to state this in the reverse fashion, we can understand something about the function of a particular cell type by considering the kind of proteins that it has available. But all of this discussion has sort of taken for granted that cells exist in isolation, and they don't. In fact, even single-celled organisms are constantly being bombarded with a broad range of stimuli impinging on them from the outside world, stimuli that influence the way the cells function. Now, we saw an example of this when we looked at E. coli and how they respond to the presence of lactose. This was a stimulus in the environment. The presence of this stimulus, lactose, causes E. coli to change the assortment of proteins they produce. And in this case, lactose acts as what we called an inducer that turns on the genes associated with its own metabolism. Another way to describe lactose in that system would be to describe it as a signal. It's a signal from the environment, a signal that it should produce the enzymes to digest the signal itself, lactose. And in that sense, E. coli is responding to the signal in an adaptive fashion. Now, by adaptive here, I mean that the ability of E. coli to detect and respond to this signal ultimately increases its fitness. It increases the ability of these E. coli cells that can do so to reproduce more. And this is because it enables the cell to specifically tailor its function to very specific present needs. Now, in multicellular organisms, the predominant environment experienced by most cells are the other cells that surround it. Thus, the cells in multicellular organisms really should be sort of seen as social entities. They're constantly talking to each other. They're constantly sending signals and receiving signals and changing the way they behave in response to those signals. These signals are molecules that are produced by one cell, released to the outside, and then go to another cell, impinge on that cell, and change its behavior. These signals can be separate from the cell, or they may actually also be molecules that are attached to the surface of the cell. Of course, it makes sense that cells having different functions, or cells occurring in very different parts of an organism, say especially a large multicellular organism, have some need to communicate. This communication, as we've said, is important in order for the different parts of the organism to coordinate their activities so the organism as a whole can respond adaptively to any external or internal stimuli. But even cells that are located immediately next to each other, even the same type of cell in a tissue located immediately next to another of the same type, needs to communicate. Think of the different cells in a multicellular organism as being like different musicians in an orchestra. Musicians that play very different types of instruments and that sit in different parts of the orchestra, such as the oboe players over here and the violinists over there, have to listen to each other and they have to respond to each other if the orchestra is to sound good as a group. But even among, say, the second violins, a group of 6 or 8 people who are all playing exactly the same piece of music, even those 6 or 8 musicians have to listen to each other, to the person right next to them, so that they can respond to each other in such a way that the section sounds like its own integrated whole. In the same way, adjacent cells in the same tissue have to communicate to function as an integrated whole. Now, signaling between cells, as we'll see in a couple of lectures, is especially important in the context of development. The signals that are sent and received by cells direct when and how much those cells will divide, how cells will differentiate into different types, and in fact how cells move around and end up in the right position in the developing organism. So for all of these reasons, we have to understand something about how cells communicate, essentially how they talk to each other. And this will be the subject of today's lecture and the next. Now, we can divide the process of cell signaling into three components or stages, you might want to call it. And we're going to use these stages as a way of organizing our discussion. So let me list these stages for you at the beginning. The first stage is reception. And this means exactly what its name implies. In order for a cell to be affected by a signal in its environment, it must have some mechanism for detecting or receiving that signal. Again, think of our analogy to musicians in an orchestra. If our violinist is going to be responding to the oboe player, she has to be able to hear that oboe player, which of course is something she does using her ears, a kind of receptor. What's the nature of the receptor, or the ear essentially, that a cell uses to detect signals from the outside? That's the first question that we need to answer. The second stage of cell signaling we call transduction. Now, transduction is a technical term that you may be less familiar with, but it simply means converting the signal from one form to another. Now, in the case of our musician, the signal that's received by the ear is a sound that's actually a physical vibration in the air. But it has to be transduced into some signal that's inside the brain of the musician. Of course, the signal inside the brain is a very complex signal having to do with the neural activities of the parts of the brain that are processing sound. Now, the reason that those signals in the brain are so complex, I should add, is because those signals have to be processed, have to be integrated with other signals in order for the musician to respond appropriately. There's more than just the sound of the oboist that the violinist might be listening to or paying attention to generally. And the same point is true when we're talking about cells transducing signals. The signal will be transformed from one form to another, and as we'll see, the transformations that can occur to that signal can be very complex indeed. Again, so that that signal could be integrated with others in an adaptive fashion. Now, the simple point to be made here, by the way, is that an external signal, at least, has to be converted into some sort of internal signal, which is analogous to the sound being heard by the musician being turned into a pattern of brain activity. It's the internal signal, both in the brain and whatever the signaling is in the cell, that ultimately will cause the response that's adaptive. So we have to ask as our second question, what's the nature of this internal signaling in cells? The third and final stage of cell signaling we need to consider is the response itself. When our musician responds to the sound of another musician, she changes her behavior, for example, by changing the way her muscles move, perhaps so that she plays a passage at a slightly different tempo, or maybe at a slightly different pitch, so that she's coordinating her playing as best as possible with the other musician. Cells respond to signals they receive in a variety of ways, mostly having to do, as you might guess, with the activity of proteins within them. This might have something to do with whether or not a protein is produced, such as we saw in the case of lactose affecting the behavior of E. coli cells. But as we'll see, it also can have to do with changing the behavior of an existing protein that's already present in the cell. So in general, the final question that we'll have to address is how is it that cells actually change their behavior? How do they respond in an adaptive way when they receive a signal? We're going to talk about the first of these three stages, signal reception, in some detail for the remainder of this lecture. And then in the next lecture, we're going to shift our perspective, our focus, to looking at the transduction of signals and then the response that cells make to those signals. Now, before we get into the details of how cells signal to each other, let me give you a tangible example that can provide a context for our discussion. Imagine you're driving down the road and all of a sudden you notice a police car turn on its lights behind you. Now, if you're like me, I imagine that seeing those lights come on, even if you're not speeding, gives you a distinctly unpleasant, startled reaction. Or if you can't relate to that example, think of any other context in which you've been seriously scared or startled. When this happens, you experience a whole suite of reactions, some that you sense directly and others that you don't. Your own subjective experience to these effects are strong feelings of anxiety or maybe excitement. But there are also physiological effects as well. For example, your heart begins to beat faster, pumping more blood to your brain and to your muscles. At the same time, blood vessels in your digestive tract actually constrict, reducing blood flow to your gut. Your blood pressure also increases dramatically. Your brain begins to consume more oxygen, which is a measure of heightened awareness. Less apparent to you, but equally important, cells in your liver, you probably haven't thought about this when you've seen the police car, but cells in your liver begin to release the sugar glucose into your bloodstream. And what this does is it increases the total amount of blood sugar and thus the amount of energy available to you. These liver cells do this by breaking down a molecule called glycogen. Glycogen is a carbohydrate that's essentially a long polymer of glucose molecules, and in that sense, it's a storage form of glucose. Now, taken all together, these various responses are called the fight or flight response. It's a coordinated response that's triggered by an immediate short-term stress. Now, this coordinated set of reactions is called the fight or flight response because it presumably evolved in the context of preparing the body to meet some physical challenge associated with danger or stress, to either fight or to run away. Your muscles receive more sugar, they receive more oxygen, less essential systems such as your digestive tract are temporarily shut down, while other very important systems such as your brain are ramped up. All of these changes presumably make you better able to meet whatever challenge you've just encountered. In the most primitive sense, as I said, to fight or flee. Although I suggest you do neither if you happen to be pulled over by a police car. Now, here's the point I'm getting to. The fight or flight response is largely caused by the action of a single hormone called epinephrine. The older British name for epinephrine is adrenaline. It's called adrenaline because it's produced by the adrenal glands, which are glands that sit just above your kidneys. So actually a slang name for the fight or flight response is called an adrenaline rush. When you're startled, the adrenal glands release epinephrine into your bloodstream, and the bloodstream quickly circulates it to all of the cells in your body. Epinephrine then is an example of a signal produced by one set of cells in the adrenal glands, that affects the behavior of other cells in the body, such as liver cells, causing those other cells to change their behavior in adaptive fashion. For example, causing liver cells to convert glycogen to glucose. Now, the question we really want to ask here is, how does epinephrine work on a cellular level? I want to use this example to consider in more detail the mechanisms that cells use to signal each other generally, looking first at how they receive the signal, how epinephrine actually is received by the cell, then how they transduce that signal, and then finally how cells ultimately can respond to the signal. We're going to focus on the effect that epinephrine has on the liver, causing it to convert glycogen into glucose, which can be used to provide more energy to a startled individual. And specifically, I want to look at this process on the cellular level, asking how that epinephrine molecule, which is a signal sent by the adrenal cells, changes the behavior of liver cells. Okay, well, let's first consider the problem of signal reception. Epinephrine, like most other molecular signals that cells use to talk to each other, can't just simply enter the cell. It can't cross the cell membrane directly. Now, we're going to talk more about the properties of cell membranes a little bit later in the course, but for now, let me tell you a little bit about the way that membranes are put together. Membranes are composed primarily of lipids. Remember, these are the kinds of biomolecules that we commonly call fats. And more specifically, membranes are composed of a class of lipids called phospholipids. Now, phospholipids are so called because they actually are sort of a two-parted molecule. On one side of the molecule, on one end of the molecule, there's a chemical group that is surrounding an atom of phosphorus. And this chemical group actually carries with it an electrical charge, a negative electrical charge. Extending off of this phosphorus part of the molecule are several, maybe a couple tails that are composed of a long chain of carbohydrates, a long chain of carbons attached to hydrogens. So you can think of a phospholipid as sort of having a phosphorus head and from which extend a couple of long carbon-hydrogen tails. Now, the interesting thing about phospholipids is that the chemical properties of the phosphorus head and these carbon tails are quite different. The phosphorus head, because it's electrically charged, is attracted to water. It's what we call hydrophilic, whereas these carbon tails are repelled by water. They're not charged, and we call them hydrophobic. Now, the significance of this is that if you have phospholipids in an aqueous medium, they actually will naturally line up in two parallel rows with the phosphorus heads on the outside towards the water and the carbon tails that are hydrophobic on the inside facing each other. The way this is commonly drawn, by the way, is to designate the phosphorus heads as little circles and the carbon tails, the carbon-hydrogen tails, as little squiggly lines extending from those circles. And you would draw then this bilayer that's created in an aqueous medium as a bunch of circles on the outside with tails facing in two parallel rows. This is the fundamental composition of cell membranes, a phospholipid bilayer that, because it has is composed of these internal hydrophobic components, makes it very hard for charged molecules to cross the membrane. So we say that the membrane is semi-permeable. Only a few things can cross it. And in general, large molecules or electrically charged molecules, such as many proteins, actually such as most proteins, can't simply move across the membrane. Okay, now that's a little bit of background on membranes, but the point of this is that signals in general have to exert their influence on cells indirectly. They can't just go into the cell and do something on a molecular level. They have to influence something on the outside of the cell because they can't get in. So how is it that a signal can impinge on the outside of the cell and then have this cause something to happen on the inside of the cell? As I've said often in many lectures, most of the important jobs that are done in cells are done by proteins. So it should come as no surprise that proteins are responsible for receiving the signals outside of the cell and somehow transmitting their effects to the inside. Proteins that have this function we call receptor proteins. The interesting thing about receptor proteins is that they're physically embedded in the lipid bilayer. Now, I described this bilayer of the lipid bilayer of the cell membrane as being composed of just phospholipids, but in fact, if you looked in detail at the lipid biomembrane at the lipid bilayer of a cell membrane, you would also find all sorts of proteins embedded in it. And one class of these proteins are receptor proteins. Now, the interesting thing about receptor proteins is that they generally span the entire membrane. They're long enough that one end of them sticks out on the outside of the cell, and the other end sticks out on the inside of the cell. So how do receptor proteins work? Well, as is usually the case with protein function, it all has to do with shape. The signal molecule, a molecule of epinephrine, for example, binds to a specific site on the portion of the receptor protein that's sticking out on the outside of the cell. Now, we've seen this before in several contexts. The shape of the protein on the outside of the cell provides a site where the physical and chemical properties of the amino acids located at that site will fit specifically with the properties of the signal molecule itself. The molecule will come in and literally sit on some receptor site that's characteristic of this protein. The significant, or the thing to keep in mind about this is therefore the interaction is highly specific. Only particular molecules can interact with the receptor protein. By the way, we call the molecules, signal molecules and other molecules that interact with receptor proteins this way, ligands, and I might use that term as well as calling it a signal. Now, when a signal or a ligand binds to the protein, the result is that the receptor protein, because of the binding, changes its shape. And the key to understanding how signal receptors work is that the binding of the signal molecule to the part of the protein on the outside of the cell membrane causes a change in shape that influences the shape of the part of the protein that's sticking out on the inside of the cell membrane. So the protein molecule as a whole, the receptor protein molecule as a whole, is changing its shape. But something causing it to change its shape on the outside of the cell can have its influence affecting the inside of the cell because the shape change is transmitted through the protein through the lipid bilayer. Now, let's look at the specific example of what happens when epinephrine binds to its receptor to make this point clear. Although cell signal receptors generally interact in a highly specific fashion, as I said, with only one or maybe a few kinds of signal molecules because of the specificity of the binding site of those receptors, actually receptor proteins can generally be grouped into just a few broad classes. In other words, there are a few basic molecular mechanisms by which receptor proteins will transmit some effect on the outside to an effect on the inside of the cell. The receptor for epinephrine belongs to the class of receptor types called G protein-linked receptors. That's a big name, but that's it. G protein-linked receptors. Now, as the name of this class of receptors suggests, they all involve, as we'll see, an interaction with something called a G protein, hence their name. And we're going to get to that point in just a minute. G protein-linked receptors are actually very large proteins. Their structure includes 7 long regions that are alpha helices, each of which independently spans the entire membrane. So this protein actually sort of goes back and forth several times, all in one clump, through the membrane. And of course, these alpha helices are connected by amino acids forming short loops on both the inside and the outside of the cell. One of the loops of amino acids that's connecting these alpha helices that occurs on the outside of the cell is where the specific site that binds epinephrine occurs. When epinephrine interacts with this site, by fitting into it, the result is a change in the shape of the receptor protein overall transmitted through these alpha helices to the inside of the cell, which results in some corresponding shape change in one of the amino acid loops on the inside. Now, what does this do? Well, there's another protein involved here, and this is the G protein I mentioned just a moment ago. The G protein is actually also a membrane-bound protein. It's physically attached to the membrane, but it doesn't span the whole membrane. It only sort of sticks into the membrane on the inside with an exposed space on the inside. The G protein generally is located quite physically close in proximity to the G protein-linked receptor. Okay, there's a lot of words there. The G protein is one protein molecule, and it occurs in adjacency to the G protein-linked receptor, which is another protein, a very big protein. Now, when the shape change occurs that's triggered by the binding of epinephrine on the outside of the cell, this shape change on the inside of the cell causes the G protein to move more closely to the receptor protein itself. This association, this closer association between the G protein and the G protein-linked receptor, in turn causes the G protein to change its configuration. Now there's a little detail because it's a little more complicated than just the G protein now changing its shape. Actually, what happens with the G protein is that it also binds yet another molecule, a molecule called GTP, which stands for guanosine triphosphate. This is a small molecule that's chemically related to the nucleotide guanine that we've seen earlier in our discussion of nucleic acids. And I don't want to go into the specific mechanisms now, other than to say that it's this binding of GTP to the G protein which essentially turns that G protein on. So the G protein is normally occurring in an inactivated or off state. And when it's in this off state, it's sort of in proximity to the G protein linked receptor, but not quite touching it. When the G protein linked receptor changes its shape, that causes the G protein to move closer, which causes the G protein to link up to have binding to it, this molecule GTP. That turns the G protein on. All right, so we've gone through a series of steps here. We've got the receptor changing its shape. That causes the G protein to change its configuration in a bit of a more complicated way. Now what happens to the G protein? Well, when the G protein is turned on, or as we say activated activated, what happens is that the G protein now physically moves away from the receptor. It moves to another molecule, yet another protein, that is also a membrane-bound protein on the inside of the cell. This third molecule, this third protein, is an enzyme. And I'll give you the name of this enzyme. It's called adenylyl cyclase. And that's a mouthful, and I'll probably mispronounce it if I say it too often in this lecture. So you can be assured I'll try not to do that. Now, the G protein then, in its activated form, is acting like a switch, because when it goes and interacts with the adenylyl cyclase, it turns the adenylyl cyclase on. How? By changing its shape. Now, I realize that this is getting to be a little reminiscent of the nursery rhyme, The House That Jack Built, or something like this, because we have the receptor protein changing its shape, causing the G protein to change its configuration, which goes over and causes this adenylyl cyclase to change its configuration. We have really what's a sequence of molecular events. And we're going to come back to this point about sequence later on in the next lecture. But the point is this. What does this enzyme, adenylyl cyclase, do? Well, as any enzyme, it catalyzes a reaction, and the reaction it catalyzes produces yet one more molecule we have to add to the mix. This is a molecule called cyclic AMP, which stands for cyclic adenosine monophosphate. And as you might guess from these names, this also is a molecule that is chemically related to nucleotides found in nucleic acids, specifically the nucleotide adenine. Here's the point. It's cyclic AMP that acts as the messenger molecule that will actually convey the signal originally received in the form of epinephrine on the outside of the cell to trigger whatever response is going to occur on the inside of the cell. Let's step back and just see what we've accomplished with all of these steps. The most important thing is what I just said, that we've got a mechanism now whereby the external signal, epinephrine, can trigger something happening inside the cell, which is the production of an internal signal. And in a sense, then, this is the initial step of transduction, a process that we're going to continue on in the next lecture. Why did we have to go through this rigmarole? Well, it was because epinephrine can't simply step in and be the signal inside of the cell. It has to get an intermediary, and that intermediary is cyclic AMP. The way this happened was through a sequence of molecular interactions, from a receptor protein to a coupling protein, the G protein, then finally to a protein which generates the signal. After this sequence of events, the cyclic AMP that's produced can carry the signal anywhere else in the cell because it's crossed the membrane. Well, G protein linked receptors are very common receptor mechanisms in cell signaling. Actually, you can find many, many other kinds of signals that are picked up this way, that are received this way by other kinds of G protein linked receptors, each differing in the particular site that they have to bind externally to the signal. There are, for example, a class of receptors called tyrosine kinase receptors. Other receptors, ion channel receptors, also bind external signals. The difference is how these other kinds of receptors cause some change on the inside of the cell. But in all cases, the external signal received creates some internal signal, which then goes on to do whatever work needs to be done. And that's what we're going to pick up in the next lecture.